totally. All right, yeah. Tell me about how you're feeling, what you've been doing, and all that stuff.
Well, since we last spoke, I've been focusing on the section banks because of like the time scheduling thing. So I've
Yeah.
been trying to get as much of it done as I could, but I still have like a good chunk of it left, and I don't know if it would be worth doing that still or just focusing more on like resting.
Yeah.
Because
Yep.
well, I was just gonna say because I know like those questions are the hardest, so they're probably gonna hurt my self-esteem a little bit.
Got it.
But I know they're like it would be important to still go through them.
So this is section bank two,
Yeah.
and
I have so section bank one, I've finished everything except for like part of biochem, and then section bank two, I finished Psych Soc, but I haven't really touched the other two. So
got it.
like still like a pretty big chunk.
Okay. All right, let's see. We can all right, and what else have you been?
I've been doing a lot of cars to try and like keep my strategy up. Um, and then I mean reviewing the section banks has taken a long time, and then I've been looking at like psych. Like I've briefly been using the psych like 300 page doc, but I haven't gotten too far into it. I just don't know like how much that would help
Mhm.
to just like read through it.
Got
Um, yeah,
Okay.
analyzing my
Yep.
strategy.
And what with your strategy?
Hmm?
Oh, and you said something about
Just just like with every questions that I do, like just kind of updating my strategy and like adding to what I want to change for the next time.
got it. All right. So I would say tomorrow to not do anything except for Anki and like perusing through the quick sheets
Okay.
that are shared. Um, and then for today, like I don't expect you to like complete, all of like the rest of the section banks, but we could go over some questions from the section bank that I can choose depending on your like so what topics are still giving you trouble?
Well, I guess stuff that I wanted to go over today was like enzymes and like the different types of enzyme classifications.
Okay.
Um, and honestly, like content wise, biology
Yeah.
stuff, I feel like I'm missing the smaller details and putting them together with the passage.
Okay. Um, so any like particular bio, like, because the thing with like BB is that a lot of people's errors can come from just like data interpretation or passage information or like experimental like design. So, uh, so are you saying that it's for you like the content stuff that might be a little?
I mean, I think there's some for all of the content, I think the worst content gap that I might still have is with bio, but then the data interpretation of biochem is an issue for me. So maybe if we could go over like a passage that has like pretty detailed graphs,
Mhm.
that would be helpful.
Got it. And so aside from the data interpretation aspect of bio, um, what type of bio content would you feel that you are that you have gaps in? Because you mentioned having some content gaps in bio.
Um, trying to think of like
Mm-hmm.
um I've been having issues with like recombinant DNA and like restriction enzymes
Okay.
and I guess just like the process of transcription and DNA replication.
Okay. Tell me what you know about DNA replication.
Um well, there's like there's a coding and a non-coding strand and like template and a non-template strand
Okay.
and um there's a lot of different enzymes involved with DNA replication. Like first the I think like the primer is put on the DNA by DNA polymerase
Okay.
and then um like there's helicase that like unzips and unwinds the DNA. Then uh, topoisomerase prevents like um, twists in the DNA like on the outside.
Okay.
Um, and on the temp or on the strand that is going from 5' to 3', I think that's the way that the polymerase reads. Um, and then on the other strand, it has to go in the opposite direction and like fill the like Okazaki fragment gaps, like in the other direction.
Mhm. So when it comes to DNA replication, does it even matter if there's a coding strand or not?
No, not for replication, but for transcription or
Yep.
there.
Good. So replicate because like you also want to realize that like roughly like 2% of DNA actually codes for stuff, and DNA replication just makes a copy of the DNA itself. 98% of that is, not coding for anything, but you're still replicating. Okay. And transcription, tell me about that.
Um, Transcription is the process of turning DNA into RNA.
Okay.
And um,
Are we like taking DNA and turning it into RNA? Like,
no, you're like transcribing
yep,
it.
yep.
Um, so the this would be the non-template strand, I think. I get confused with the names of like coding and the sense and the anti-sense
Yep.
strands.
So sense is going to be the coding strand, and uh antisense is going to be the template strand,
And
and,
the template strand is like complementary to the mRNA that you're making.
mhm, as well as the coding strand, right?
Hmm.
So that's why it's like, yeah, okay, okay. Tell me, so we transcribe the DNA into RNA. What do we what else happens?
Um, well, when it's being transcribed, the complementary base gets added to the
Mhm.
RNA. So, like cytosine if there's a guanine, and like adenine if there's a thymine. Um, and what else? Um, I guess like when they get well, there's like a lot of other stuff too, but now I'm blanking.
So RNA, is that like a very stable molecule?
Um, I'm gonna say no.
Okay. So Uh, so RNA is something that you want to just create or transcribe if you need to make that protein, if you need for it to be translated into that protein.
So
Uh,
there would be like
huh?
transcription factors that like determine if it's going to be transcribed or not, or like depending on the need of the cell, I
Yeah.
think.
So a transcription factor can be a like a enzyme that can bind to the DNA inside of the nucleus and cause transcription of the RNA. And you can't take the DNA out of the nucleus, but you can take the RNA out of the nucleus. But the RNA is very, very unstable because of two things. So, one, if you just think about the structure itself with the ribose sugar, the pentose sugar, right?
Mhm.
Um, for DNA, there's just that 3' hydroxyl group. But for RNA, you have the 3' hydroxyl group as well as the 2' hydroxyl group. And what do I always say about hydroxyl groups?
That they can be phosphorylated.
Uh, more? Let me, more?
That they're good nucleophiles.
Yes. So, yeah, predominantly think that like, because they're good nucleophiles, they can be phosphorylated, right? So,
Mm.
yeah, in ribonucleic acid, you have two hydroxyl groups next to each other, and they can have like a nucleophilic reaction with it's like an intramolecular one that can form like an epoxide. So the molecule intrinsically, like the ribose molecule is intrinsically less stable than deoxyribose. In addition to there being exonucleases in the cell that would, what do you think they would do?
Like leave the cell or well,
What's
I
a
think we
nuclease?
talked about this last time, but like endonuclease and exonuclease have different like meanings than the typical.
Oh yeah. Or maybe we were talking about
Oh no, that was
toxins,
yeah, that was endo.
toxins. Uh,
I'm guessing exonuclease like leaves the nucleus
so a nuclease would do what?
or break down nucleotides.
Yes. And you know about like the restriction fragment, like the restriction endonucleases?
Mm.
They would what would they do?
Um, they like degrade specific parts of the um strand
Mhm.
so that like it can be added somewhere else.
Yep. So like it could be in like a palindromic sequence that can make sticky ends that can then come back. So okay. So if so an endonuclease would break down a nucleic acid like someplace in the middle, and an exonuclease will break it apart at the ends. So that's why when RNA gets, when DNA gets transcribed into RNA, the RNA would have a step where they add like a 5'
Mm.
to protect the 5' from exonucleases, and then a poly A tail on the 3' end or whatever to protect it also from exonucleases,
Hmm.
right? And then they do one more thing before the RNA turns into the mRNA.
All like alternative splicing.
Yep. Tell me about that.
That's when the introns like stay in the nucleus, but the exons get like spliced together, and then that the like mature RNA travels outside of the nucleus.
So tell me again about introns and exons.
The exons are like part of the mature mRNA,
Okay.
and the introns like get cut out and they like stay in the nucleus
Yeah,
versus
yeah, or whatever. You throw them out, you don't need them, right? So
yeah.
the exons are going to be like the coding parts of the gene, and so that's splicing. What's alternative splicing?
Like picking and choosing which exons you want to or the molecule like puts together.
Yep. So you can have like let's say exons 1 through 4. You could end up with, let's say, exon 1 followed by 3 followed by 4, but you could also end up with exon 2, 3, and 4. So you have a lot of variation there, and that can make protein isomorphs, which are versions of the same protein with slight differences based on the differences in exons that compose the mRNA that codes for that protein.
Hmm.
Good. So I'm putting a question in the chat, and you could like, yeah, like share your screen if you want to show this, so I have something to, like write on. But yeah. And if you've done this already, like let me know.
No, I haven't done this one.
All right, go for it.
Um, the arrows, is that showing like what are the arrows versus like the base pairs? Because like the arrows are going in opposite directions of the base pair. Uh, well, I'm not sure, but the exogenous protein, like maybe that's important.
Yeah.
Um, I'd maybe put it like between 396 and 454.
Mhm.
Um, I
Yep,
don't know what a polylinker is, but I'm guessing that's like links multiple proteins together.
yep.
With
So there's something in Star Trek called the Kobayashi Maru like thing or whatever. And it's basically like a simulation in which you are in an unwinnable scenario like situation. And like I guess the theme of it is like how will you react to things that you don't know? So here we have like what you were saying about yeah, you were like, okay, what do like the arrows mean? Um Polylinker, etc. And I wasn't like helping you out or anything like that. But you chose B. Tell me why.
um, well, I chose B honestly because it's like it seems like it's the only area that's like more important than the other areas.
Yeah.
Um, but also like they're trying to express a protein. Um, so I don't know, like I just thought polylinker might have to do with that.
Yeah. So, for instance, 0 to 350 is like here to like here, right?
Mm-hmm.
1000 to 1500 is here. 2000, I guess, like over here to 2500 is here. So
Mm-hmm.
these are all regions that include these black arrows. The only region that has like that's not like exactly the same as the other ones, meaning like, you know, this kind of white region here is the one that's the most different, right?
Yeah.
So that can be a clue. And also, if you look at, okay, there's 350 base pairs here, right? There's a 500 here, there's 500 here. Here, there's like what, 58
Mm-hmm.
for this one? So, so yeah, I wanted to give you a scenario where you, you know, like so basically, you're gonna try to if you look at a question, first step also always to make sure you understand what the question's asking. And unless it's like directly referencing a passage, like you're gonna try to use your knowledge to answer it. But if you completely don't know about what, the things mean, then you can at least rely on your like critical thinking skills, which include uh pattern recognition and like deductive reasoning.
Mm-hmm.
So good, 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 good. Um, okay.
So like with this question, was there any like content that I was missing to answer this, or they kind of like put it there because it's confusing?
Yeah. So I mean, like, of course, plasmids are on the test, and
Yeah.
we need to know about them and all of that stuff. And oh, and then these arrows are genes, right? And
Mm-hmm.
um, I think ORI is like the origin of I think replication or something like that. Like if this plasmid starts to replicate itself, it starts there at like perhaps 1500 or 1000 base pairs or whatever. Um, and you could also, I guess, see that it's like 0 to 500 to 1,000. Like the plasmid uh is going forward like clockwise, and so counterclockwise is like the, I guess, the replicated you know um like gene or DNA going in the opposite direction, like I guess like 3 to 5 or something. But yeah, there isn't really, I mean, like, yeah, there's plasmids on the test and, etc. But something as specific as a polylinker, I don't think you're not really expected to know that. So, but yeah, I just really wanted to have you use your critical thinking skills, which you did perfectly. Um, okay. Let's see if I'm gonna try to find another one. As I try to find another one, you can kind of tell me if what other types of things that could be useful.
Mm, anything having to do with metabolic pathways
Hmm.
would be helpful.
Okay. And
I
yep.
Tomorrow I'm planning on reviewing the like specific enzymes and like where the energy molecules are with each
Mhm.
step, but I still like get them all mixed up.
Yeah, yeah. So that could be something where you can have like a big picture kind of understanding of like what, and also like in the PDFs folder I have lots of like the metabolic pathway notes, so, and some of them include like what's the purpose of it and what's the location of it. So yes, that can definitely help. And you mentioned like the enzyme classes, and let me see if I can find another um thing. Okay. In the chat.
Um, I'm gonna say 50%.
Mhm.
Um, I was between 25 and 50,
Mhm.
but I think I'm sticking with 50.
Good job. Okay.
So, like in PCR, first you have like the two strands, they get pulled apart, and then those two get replicated.
Okay.
So they would have, um, they would have one, well, hold on.
Mhm.
So it's saying that the radioactive molecule or the phosphate is on both of the strands, so they're both radioactive. They replicate and they make one of the other strands that's not radioactive,
Yeah.
or that, yeah, that's not radioactive. Then that breaks up again
Hmm.
and makes, well, hmm, yeah, I'm still a little confused on how that works, I think.
Sure. So, let's see, non-radioactive nucleotides. So let's say this is the DNA and black is non-radioactive. But now this is in an environment that's full of radioactive nucleotides. So let's see, and they're saying 2 rounds of this PCR. So let's just kind of like, replicate it. So then you get like your black non-radioactive strands that are separated like this. And then let's say that blue is going to be uh, the radioactive nucleotides that start to form for the next strand in the like yeah the products here. That's after 1 round of PCR. And then for the second round, you'll have, again, one black strand for each. And then you'd get that. And oh yeah, for this one it's blue and blue. Then here it's blue and black. Then here it's blue and blue. So they're asking what percentage of the DNA molecules will contain the radioactive phosphate on at least one strand. One.
Wouldn't that be 100%?
Oh, wait, right. Hold on.
Because when I was reading it, I was confused as to like, it's saying that they're using non-radioactive nucleotides, but the template is radioactive. So, like, isn't the template what is starting out as radioactive? And then as it's replicating, it's adding things that are not radioactive?
Yeah, I read this wrong. I thought that the only thing that was radioactive was the environment. Okay, let me do this again then. So, um, okay, so yeah, I got it the other way around. So the nucleotides that are like free are non-radioactive, but the DNA template itself is radioactive on both strands. And we're not really differentiating like template strand versus coding strand or anything like that here because we're really just replicating and you don't really need to differentiate between coding and non-coding if you're just replicating. So, but yeah, on both strands, they tell us. So okay, so I will start off with blue indicating radioactive. And then when this unwinds, you would get the blue radioactive strands. Now, see, so okay, now the environment does not have radioactive nucleotides. So the other strand for this is just going to be black, and that's the color I'm using for non-radioactive. And then when this unravels yet again, we get a black one, a blue one, a blue one. And then since the environment has the non-radioactive nucleotides, that means a black one comes here, comes here, comes here, and here. And yep, 50%.
Okay.
Yeah. So I read it as DNA that we start off with, non-radioactive, environment, radioactive, which would mean it would incorporate radioactive nucleotides into the DNA through replication. But yeah, you're right. That would have been 100%. So then I reread it, and it's really that the environment has non-radioactive nucleotides, but the template we start off with will be radioactive.
Okay.
Hmm. So is this kind of like how you were doing it, or?
Yeah, it was how I was doing it, but I was like getting mixed up at the end. Um, like if the strand that gets replicated from the radioactive portion
Yep.
would become radioactive.
Mhm.
So, but um it wouldn't, so
Yeah. Okay. So let's have you open up the Jack Weston MCAT quick sheets. That's in the um yeah, in the shared drive. And we can kind of look over certain topics like the metabolic pathways and stuff like that. Yeah, it'll just be in the folder that you're in right now.
okay.
Uh the previous one. I mean, like, yeah, not in the PDFs folder.
Oh, not in, okay.
Yeah. So if you just scroll down, yeah, there it is. Okay. So we can kind of yeah, like peruse through this, or we can actually go right to like the metabolism stuff. So where is maybe carbohydrate metabolism one
Okay.
could possibly help us. So well, I guess I could also just ask you like, you know, tell me everything you know about glycolysis, perhaps.
Um, okay. Well, glycolysis is the breakdown of glucose into pyruvate, and um there's like energy consuming steps. Like the first five, I think, are energy consuming, and then the next five are energy pro like a net energy producing or energy producing, but there's a net energy production of glycolysis in general. Um,
Okay. The
yeah.
the steps that you're saying are energy consuming. Tell me tell me about that.
Um, they like not all of the steps, but like in the first half of the um process, like there's two steps that turn ATP into ADP, and then in the second half there's two there's two steps steps. Yeah, there's two steps that turn ADP into ATP, but by that point, it's like two separate molecules, like they branch off, so there's a net production of two
Yep.
ATP.
Yep. So in the steps where ATP turns into ADP, what type of enzyme would
Uh,
we?
a dehydrogenase,
So what would a dehydrogenase do? Or what class of enzyme would that fall under?
redox,
So like yeah, oxidoreductase, right?
or yeah.
But
Um,
redox, yeah. So now ATP to ADP, is that redox?
no,
Okay. So
it's a transferase,
it's a transferase. Like,
like
yep.
kinase.
So you have your gamma phosphate from the ATP taken by STY of the kinase and then given to the substrate. So what's that first the first one? Oh, so also, all right, how many irreversible steps are there in glycolysis?
Free.
Great. And what else can you tell me about them? Like,
Um, the the first one is with like glucokinase or hexokinase.
what's the difference?
Um, glucokinase, I think, is specific to the liver, and hexokinase is not, or I got them mixed up.
Uh,
One of them is specific and one of them is not.
yep. So, um, yeah. So hexokinase will do what?
It will transfer the phosphate from ATP to um or from it will transfer the phosphate from ATP onto glucose
Mhm.
um in cells other than the liver,
Okay. So like predominantly like muscle cells
muscle
and
cells.
stuff. Now what does it create? Like from
It,
glucose to what?
it glucose 6 glucose 1 phosphate or glucose 1.
Uh, so uh, so glucose 1 phosphate would make sense like if you were to polymerize glucose into glycogen because you can make bonds between the first carbon of a glucose molecule and the fourth carbon of a different one. You could also have between the first carbon of one and the sixth carbon of the other for the branch. But that would be if you're doing something anabolic like producing glycogen. But for something catabolic like glycolysis, we're gonna phosphorylate it and you're gonna get glucose 6 phosphate, right?
Okay.
And that's carried out by hexokinase, which is present in most tissues. Most likely, though, you'll see that it would be like in muscle or something like that. Which so yeah, we know that that phosphorylates glucose. Uh, in the liver, though, um, what usually happens like in the liver in the context of what we're talking about?
Um, like en energy usage.
So like in the muscle you could have like glycolysis, right?
Um,
In the liver you could have
where you can produce glycogen.
yeah. You could also produce glucose like gluconeogenesis, right?
Mhm.
So since the liver is going to be like gluconeogenesis rather than glycolysis, your glucokinase, uh, which is similar to hexokinase, uh, but is different because glucokinase has a much lower affinity for glucose.
Okay.
So that phosphorylation of glucose to glucose 6 phosphate is is not going to happen much, which is what we want because that's we want gluconeogenesis. So we want glucose 6 phosphate to turn into glucose rather than the other way around,
Hmm.
right? Good. So that's the first step of glycolysis. The first, well, yeah, it's the first step, but it's also the first irreversible step. What's the next irreversible step?
I think it's with PFK.
Okay. And tell you about that.
And that, that is also an enzyme that transfers phosphate group.
All right.
Um, I don't remember exactly which uh, molecule it's transferring to. I'm guessing it's fructose 6 phosphate, and it gets an additional um, phosphate to be fructose 1,6 bisphosphate.
Absolutely perfect. Good. Good, good, good. That's phosphofructokinase. Takes a phosphate, the gamma phosphate from ATP, gives it to fructose 6 phosphate to make the 1,6 bisphosphate. So yeah, now we have, two uh, phosphate groups added to our molecule. Now, and yes, you can connect the thoughts of like if we are having like an ATP turn into an ADP, or well, this won't happen all the time, but or sometimes you can have an ADP turning to an ATP, and that's going to be done by largely by kinases, uh, which transfer phosphates from like organic molecules to organic molecules rather than from inorganic, like a phosphorylase or a phosphatase would do. So yeah, so you're gonna have one last irreversible step in glycolysis.
Um, is it the like carbox? No. Um, does it have to do with like phosphoenol pyruvate?
Maybe.
Is it carb?
Yeah,
Is it
yeah, it does
carboxykinase?
it does have to do with that.
Okay. Um, is it like pep carboxykinase? Is that even an enzyme in glycolysis?
Um,
Or is that a different pathway?
doesn't ring a bell because the word carboxykinase
Or
might
like
mean.
carbo?
Oh, what were we saying?
I don't know.
Let me see. Because carboxykinase could then mean perhaps oh, okay. So this is in a different direction because you're making the PEP.
So like during gluconeogenesis. Okay.
Yeah.
Um,
But um but yeah, it's gonna be so the last irreversible step in glycolysis is gonna be PEP to pyruvate, and that's gonna be using pyruvate kinase, which in this case, instead of it taking a phosphate from ATP to give it to a substrate, it's going to take a phosphate from the substrate and give it to ADP to make ATP.
okay.
So those are the three irreversible steps. They all deal with kinases. So the first one, uh, and by extension, that means that they deal with phosphorylation. So first one phosphorylates glucose to glucose 6-phosphate. Second one phosphorylates fructose 6-phosphate into fructose 1,6 bisphosphate. So we've added phosphate, like two phosphates, and then the last one we remove one of the phosphates to get, well, we remove the phosphate to get pyruvate. So
Mhm.
yep, so that's those are the irreversible steps of glycolysis, and it's good to know the irreversible steps. Um, what would the delta G be for the irreversible steps?
Negative
Yep.
because it can't go backwards. Like it's spontaneous.
All right. Now, if for some parts of the reaction like let's say DHAP to turn into like or yeah, for like one of the reversible steps like fructose 1,6 bisphosphate that can turn that into DHAP, which can then be turned into like G3P, and then that can move further down. These are not irreversible steps. They could be reversible. What would make them, move that reaction forward?
Like having a lot of the reactant
Yep,
would push it towards the product.
yep. So Le Chatelier's principle, right?
Mm-hmm.
Good. Good. Okay, so yeah, for glycolysis, the three irreversible steps, the enzymes involved, the substrate and product that are made also involved. Good, you got those down. Now, um, so now the kinases, we know what those do. What other kind of reactions take place in, let's say, glycolysis?
There's an isomerase
Okay.
and mm, an aldolate.
So there could be those certainly do exist. Um, and let's say so think about I guess a type of enzyme that would be well like yeah, try maybe like think of like another like one or two. Or if you maybe think about how you would like how to describe like glycolysis big picture wise.
Like you're moving around a bunch of phosphates.
Sure. Um, but What else is happening?
Mm. Like you're dividing into two molecules.
So you're breaking down glucose,
Mm.
which is a blank type of reaction.
Hydrolysis,
Um, like,
catabolism,
yes. Tell me, so what's catabolism?
like the breakdown of a molecule.
Good or good? Um, and it's carried out usually through a type of reaction.
A lyase
Or,
or
yeah.
um
So
a,
think about, I guess,
hydrolase-
yeah, what's up?
a hydrolase.
So I guess think about when you are catabolizing something, aka, breaking something down that's like from food or something like that. Where do they what happens ultimately?
Um,
Well, I guess that was like a really vague question, but uh, so I guess what I'm getting at is that catabolic processes often are oxidative, which means that there must be some type of oxidoreductases in these pathways, like in glycolysis.
mhm.
And what could those enzymes be called? Like, what could there be, like, in the names of them? Like, what might be a name that's consistent among them? And also, what do they,
Dehydrogenase,
yeah, what's up?
a dehydrogenase.
Exactly, yeah. So, and so if there's dehydrogenases, then, what do
Then,
they do? Like, what do they take in and what do they
um,
pop out?
there's probably like an electron carrying molecule like
Yep.
NAD plus
Hmm.
NADH.
Yep. So all catabolic processes will involve oxidation, which would involve the reduction of the mobile electron carriers. So you really could think about all of catabolism as electrons being moved from substrates to, like, NAD to make NADH, FAD to make FADH2. And those just carry those electrons until they go all the way to the electron transport chain where they finally give their electrons to oxygen.
So all of the processes are oxidative, which means that like NADH would be oxidized to NAD plus, or like FADH2 would be oxidized to FAD
So
until the electron transport chain.
so the substrates are going to be what undergoes oxidation, which
Oh.
means that the, like, let's say we had Na plus, right? The Na plus is gonna, sorry, what are we, not Na plus, NAD plus, right? That's just sodium. But NAD plus to NADH is undergoing reduction, right? So
Mhm.
it gains electrons, but the thing that it's acting on, like the substrate, is going to be oxidized. So the substrate is going to be losing electrons to the NAD plus or FAD.
But then in the electron transport chain, the FADH2 gets oxidized.
Hmm. Yeah.
Okay.
So, like, all throughout the cell, you will have NAD plus and FAD act alongside oxidoreductases like dehydrogenases to take electrons. Okay. So it's like every step when you cause oxidation to substrates, you take electrons from them and you give it to NADH, FAD, and those you just build up a pool of a bunch of those NADHs and FADH2s that are holding on to electrons, so that ultimately in the electron transport chain, uh, they can finally get themselves oxidized, aka NADH to NAD plus, because they are ultimately giving their electrons to oxygen at the
Okay.
very end. Yeah. So good. So that means when we have, well, kinases, we're going to have something with either ATP turning into ADP or ADP turning into ATP. And the three irreversible steps in glycolysis involve kinases. But you will also see in all of these reactions redox, and they're called the enzymes that are frequently used, that are the oxidoreductases, are dehydrogenases, and that's when you're going to have some type of like Na plus, NAD plus to NADH or FAD to FADH2, something like that that holds on to the electrons until it gives it ultimately to the oxygen.
Okay.
Good, good, good. Now, PEP to pyruvate, last irreversible step. When it's done that, what can happen? Like, yeah, what can happen?
The pyruvate can go to the pyruvate dehydrogenase complex.
In um what situation would make it go there?
Um, like if the cell needs energy,
Um,
like if it wants to go into the citric acid cycle.
so what would determine whether it goes there or not?
Um, if you're under uh, aerobic conditions.
Okay. So if you're under aerobic conditions, what would that mean? And then what would that lead to?
It means that oxygen is available to be like in the electron transport chain as the like final electron
Yep.
acceptor.
So
Uh,
mhm. So if oxygen is not present, what would happen?
Pyruvate could be turned into lactate or
Okay.
like lactic acid or ethanol, depending on if it's in like
Yep.
yeast.
Good.
Um,
Good. Yep. So
like through fermentation.
mhm. And so pyruvate to lactate, um, what type of enzyme do you think that would be?
Um, um, transferase.
Let's see. Do
I
you
need to
know, like,
refresh on. I need to refresh on the structure of lactate, I think.
got it. The structure of pyruvate, does that like is that something that you could like what that would look like, the molecule?
Uh, I think there's a carbonyl group
Oh,
on it somewhere.
okay.
Um, is it symmetrical?
Um, I mean,
Not
maybe
really.
not, like kind of, um, but it's an alpha keto acid. So that means you're going to have a ketone in it. And what else?
Um, a carboxylic acid.
Good. So it would look like this.
Mm-hmm.
Because we have the keto and the acid part. And now lactate is going to look similar, except you get this.
So it pyruvate is reduced, um, like to turn the ketone into a hydroxyl group.
Yeah, very good. So it seems like you remember like that biochem redox, like generalized thing. Like a ketone can be or an aldehyde can be oxidized into like what?
An aldehyde can be oxidized into uh carboxylic acid.
Yep. Good. And it can be, yep, reduced to,
It can be reduced to a hydroxyl.
yep. So yeah, so this bottom one is what lactate looks like. So and the top one is what pyruvate looks like. What's yeah, what's happening between like in this arrow that I drew, like from the pyruvate to the lactate?
Um, I'm guessing there's an NADH molecule that's getting oxidized to NAD plus.
Very good. Why do you think this happens? Like why I thought we were making NADHs to give to the electron transport chain. Like why would it do this instead?
It does this to regenerate the NAD plus so that it can go back into glycolysis,
Very good. Very, very
keep
good.
regenerating
Yep,
energy.
good. So, all right, yes, and this is what happens when you don't have oxygen present, aka anaerobic conditions, and the lactate can travel through the blood to enter into the liver Cori cycle, and in the liver, the lactate will turn back into the pyruvate, but then the pyruvate will turn back into glucose through gluconeogenesis, right?
Hmm.
All right, good. So now if oxygen is present, um, what happens then?
Um, then the pyruvate goes to PDH complex
Where's
so
that?
that that's in the cytoplasm.
Maybe.
I think.
Oh, last I forgot to ask this. So this step here, what type of what do you think the name of like the enzyme would be? Or what do you think would be in the name?
Dehydrogenase.
Good, good, good. Okay.
Lactate, is it lactate dehydrogenase?
Hmm.
Okay.
Awesome. So okay, back to the pyruvate dehydrogenase complex?
Um, is it in the cytoplasm?
Maybe maybe it's inside something that is inside the cytoplasm.
The mitochondria.
Uh-huh. Good. So
Um,
your yeah.
okay. I thought it was in the, I don't know why I thought it was in the cytoplasm, but um,
Mhm.
so it turns pyruvate into acetyl-CoA
Okay.
so that acetyl-CoA can be used in the citric acid cycle.
Okay. Um, do you remember what enzymes were involved?
There's three enzymes.
Good.
Two- There's pyruvate dehydrogenase.
And one
There's what?
oh no, go ahead.
Oh, there's there a transacetylase,
Yep.
and then there's also there's another one of either dehydrogenase or transacetylase.
Mhm.
Can't remember. I think a dehydrogenase maybe.
Yeah, so you have your pyruvate dehydrogenase, which um reduces oh no, wait, oxidizes the like this part here of the pyruvate, so it leaves as CO2,
Okay.
right? Because remember, if you oxidize a carboxylic acid, it can turn into CO2,
Mm-hmm.
right? So there's that enzyme. So there's a pyruvate dehydrogenase that causes like oxidation of pyruvate so that it loses the CO2. Um, and that can easily be confused with a decarboxylase. Because a like if I see something that produces CO2, I'm first thing I'm thinking of is decarboxylase. But it's not a decarboxylase, it's just an oxidation. So that's going to be a pyruvate dehydrogenase. You can
How
also,
can you tell,
yep.
sorry, how can you tell the difference between the two then? Like if they both, like would you have to look at the oxidation numbers or like how would you tell?
Oh, so if it produced a so if it's if okay, so a decarboxylase will always produce a CO2 that will leave the molecule. Whereas a dehydrogenase is going to participate in some type of redox. So if we're going from pyruvate, which has, it's over here with a ketone and a carboxylic acid, and we oxidize this, the ketone can't be oxidized further, but the carboxylic acid can be oxidized further into a CO2. And it just so happens that that's what happens here to cause the CO2 to be released. So okay, I guess the question was okay, like
Like if there's a CO2 being released and it's a dehydrogenase, how would you be able to tell that it's a dehydrogenase, not decarboxylase?
yeah, okay. It's a good question. So let me think. Um Okay, so well, actually, yeah, I'm getting like conflicting answers when I'm like asking like ChatGPT and stuff because and then also when I look up, uh like the like PDH, like sometimes people write decarboxylase, sometimes people write dehydrogenase, which is totally weird. So I think that in general, if you see like this isn't really going to be that good of an answer, but if CO2 is leaving as like a, product, and if the reactant that loses the CO2 had or was originally with a carboxylic acid, then we can say that it could be like something like a dehydrogenase, a redox reaction. Um,
Mm.
but if we have CO2 leaving and the initial one reactant or whatever does not have a carboxylic acid, then a decarboxylase could have been the one that did that.
So in terms of like the
Mhm.
MCAT, like we're assuming that the like pyruvate dehydrogenase complex is dehydrogenase. It's not a decarboxylase.
Uh, yeah, so that can help with the memory aspect of it. Like, if the name is pyruvate dehydrogenase complex, we know that it's going to use pyruvate dehydrogenase to lose the CO2 rather than a decarboxylase, just from the name. So
Mhm.
I think that's the best I have for that. So, but yeah, so you're gonna have your pyruvate dehydrogenase, which is what the complex is named after, and that's gonna oxidize the pyruvate, resulting in the release of CO2. You're gonna have a transacetylase, which is gonna be responsible for taking an acetyl group and moving it from something to something else, and then you have a another dehydrogenase, which is what's the other dehydrogenase called? It's like dihydrolipoyl, something like that.
Yeah.
Um, and that one's going to be like a regular dehydrogenase, in that it's going to be taking NAD plus and turning it into NADH. So yeah, so for that, basically, yeah, your um yeah, so you're gonna have your pyruvate dehydrogenase, you're gonna have your transacetylase, and one last dehydrogenase. And that last dehydrogenase does what you would expect from a regular dehydrogenase, which is, some type of redox involving like NAD plus and NADH. So, uh, yeah, so that's what I would say for that.
And would you recommend like memorizing the cofactors involved, like in the complex, or is that not really as important?
Uh, I guess I would say if you understand the rest of Krebs cycle and gluconeogenesis and like beta oxidation and those stuff, then I would say like you could use this time that you have to learn, maybe just make like an Anki card for like the cofactors in the pyruvate dehydrogenase complex, because I have seen that come up before. Um, but if you're like struggling with like gluconeogenesis, like what happens, where things happen, and beta oxidation and all of that stuff, then your priority should be more so leaning towards that.
Okay.
But yeah, like, and in the PDFs folder, I have lots of like my notes on metabolic processes. So there's like the one where it's a big OneNote PDF with multiple pathways all in the same thing, like in different locations. Um, and you can kind of like, you know, understand like the irreversible steps first and the type of reagents at those irreversible steps and the types of enzymes involved as well. And then like for the pyruvate after the pyruvate dehydrogenase complex, it's going to turn into the acetyl-CoA. And that acetyl-CoA, a lot of things can turn into. Like we're talking about glucose turning into acetyl-CoA, but proteins can also turn into that. Fats can also turn into that, and they kind of all converge at acetyl-CoA. So like if you had a question asking like how much ATP can be produced from like palmitic acid, something like that, and you do the beta oxidation where you each round you cut off like two carbons,
Mm-hmm.
and those produce acetyl-CoAs, and then you can like treat them as the acetyl-CoA that goes into like the Krebs cycle and ultimately electron transport chain. Those can kind of come back together. So yeah, I would yeah, look through like the PDFs in the PDFs folder for metabolism. So you just have all of that, right? Like all of that down. Yeah, and then like in terms of importance, it's like, yeah, like glycolysis, know the like for all the pathways, like, I would say well, first, what's like know what the purpose is. Second, where it happens. And then third would be like those irreversible steps, like what types of enzymes do those entail, and what types of like reagents or products that get taken out that can happen.
Mm-hmm.
So and know that for like, yeah, glycolysis, Cori cycle, lactic acid, fermentation, and then gluconeogenesis, and then TCA cycle, like pyruvate dehydrogenase complex, TCA cycle, electron transport chain, and like also like they like to ask like what if we uncouple the ATP synthase of that? Uh,
Mm-hmm.
and then look over the beta oxidation and what else is there? Oh, pentose phosphate pathway is also important because it's actually an anabolic like process, but it's still oxidative. So, and if you just know that the pentose phosphate pathway, like what it does is it produces those like ribose sugars that can be incorporated into like nucleotides and things like ATP and GTP and stuff.
Okay.
Hmm.
So then, like, I'll focus on that tomorrow, and then you would recommend like going through
Hmm.
the quick sheets,
Yep,
I
yep,
guess.
yep. So like, yeah, like the oh, and like you remember like the what is it? like osmotic pressure versus hydrostatic pressure, that kind of stuff.
Yeah, I'm like gonna go through that again just to like grill it. But
Got it. Yep. So yeah, I would just go like yeah, peruse the PDFs folder where I sent you like a text about like the pathogens thing.
yeah,
Yeah.
yeah.
You can look at those. And yeah, so look at the PDFs, particularly like, the metabolism ones, but you can look through all of them. You could, yeah, go peruse through the that quick sheets like PDF. Um and what else? And yeah, and I guess like yeah, for like tomorrow just do like, don't really do like questions, but like just kind of yeah, like do like more of like a passive review, do like some Anki, look through that quick sheet,
Okay.
write stuff down that you feel like you'll forget that can help you. Um, and oh, and then like, yeah, if you feel like anxious and stuff, like during the test, like, just give yourself some time to like breathe. And we talked about like what to do during like your breaks and everything like that. So you can do that. Um, and try to go to sleep like 2 hours earlier than usual because people always take longer to sleep beforehand, on
Mhm.
the night before their test, so we can anticipate that. And then yeah, at that very end, like when I say very end, I mean if you don't know like the answer, and your knowledge isn't helping, the passage isn't helping, um yeah, just remember it's a critical thinking test and that you can figure stuff out without having to know it all as I mean like somewhat of a last resort, which would be like how you handled that polylinker question that we just did, which is at the end of the day or whatever, if you don't know like what that stuff is, will choose you'll use your, critical thinking skills to choose the one that is like the most different, right?
Mhm.
In, more than one way. So like, yeah, so you'll always be able to like, if you need to, rely back on your pattern recognition and deductive reasoning. Okay.
Okay.
Awesome. So yeah, I think you're gonna crush it because you've been getting these 512s like always like, over and over again. So you're probably gonna, you know, at least have like a minimum of that. Keep you know, yeah, keep an eye on like time, using those checkpoints, learn to like I mean, you already have to like do timing and all that stuff. So yeah, I think you're gonna crush it.
Thank you for your help.
Yeah,
Like,
of
you're
course.
really
Yep, of course. So yeah, if you have any other questions,